So any questions from last Thursday or Tuesday or anything we've covered thus far? Everyone's like, we got this. It's pretty easy stuff, right? Maybe? No? Okay. It'll become easy in time. Just wait. All right, so we were continuing our talk on enzymes and how we get energy out of the body, at least with what we put into it. We mentioned that in a lot of cases you're going to find that these processes there's multiple enzymes that are involved. It goes through kind of a pathway. So you can see here how, start out with your initial substrate A, and then through a series of different enzymes, you're eventually going to have these intermediate products, which may not be entirely useful to us, but then you get your final products. That's one particular way we could do this. Is if you have kind of multiple or branching pathways. You can have different ways to skin the same cat, essentially, where you can start with your substrate, and then depending on kind of where you're at in the chain, you can have different enzyme processes that can occur here to eventually get your final products. We see this a lot, at least from the pharmacy world, because drugs can have different metabolic pathways to detoxify them and get them out of the body. So a really good example of that is acetaminophen, which you mentioned the brand name for that is Tylenol. Absolutely. So Tylenol gets, and you guys know like what major organ takes a hit if you have too much Tylenol in your system? The liver, right? Everyone kind of knows that. So it's pretty famous example of a liver-induced or drug-induced hepatotoxicity. But basically, there's several different pathways that acetaminophen can be metabolized in order to get eliminated from the body. In some cases, you may go through a sulfation pathway, which are enzymes involved with that. In some cases, you may go through a glucuronidation pathway, glutathione conjugation. So there's lots of different ways your body detoxifies it to get rid of it. But then when you overwhelm those processes, that's where you can have problems that can come about, and that's where you can get the liver damage that happens. We'll talk about that more in pharmacology in the upcoming semesters. But just an example of how there's different ways to eventually get to the same end state, basically. So again, you can have negative feedback loops that get involved in this process as well. So for instance, if you were to have a pathway that ends up, when you get the different final products, so either F or F prime here, you can end up having accumulation of one of those products, and it goes back and feeds back to stop further substrate going through that pathway, right? So sometimes you can have some feedback inhibition going through that as well. In which case you just have the other enzymatic pathway being favored at that point, and you have more of that final product being made there. Now, have you guys heard the term inborn errors of metabolism? What does that really mean? Pretty much what it says, right? Inborn just means that it's usually a congenital abnormality that occurs here. Basically, if you have a mutation, either in a single or multiple genes, that would have coded for an enzyme, maybe they don't code for that enzyme anymore, or maybe it's a defective version of that enzyme, or maybe just not enough of it. Basically, that's going to start to kind of throw a monkey wrench in the works, and those different pathways are going to become very much altered. In a lot of cases, you can find that this may not be compatible with life in a lot of cases, or they may have very significant kind of outcomes about this. Either, we see this quite frequently over at Nemours, especially because you normally are going to catch these in either like kind of the neonatal period or as their children, because it becomes more apparent as they start to have these processes that occur. Usually they have like mitochondrial deficiencies where they can't produce certain products appropriately, and so they can have all kinds of different issues that pop up there. Probably you'll get more of this in your genetics course, but just realize that you can't have a congenital issue to where you're not producing these enzymes in the correct either form or number, and that can lead to all kinds of different disease states. And they get really, really odd because you have different things where I have to give citrulline, because the body can't produce enough citrulline for these patients, and so we're actually having to get these kind of bulk powders. They're not actually really drugs per se. They're kind of just there's an amino acid they can't produce themselves, so we're going to supplement by giving them these amino acids. In some cases that's useful because we can help to kind of supplement those pathways and give it to them, but in other cases we may not be as successful. It just depends on where the error is actually occurring. There's just so many multitudes that are out there. That's definitely a big use for our genetics department, is where we get those guys involved to kind of help us determine what mutation has occurred, what products are not being made, and how do we treat that patient effectively. So very, very complicated, but if you're interested in it, there's lots of resources out there for that. Okay, so again, just to give you an idea what this would look like, you can have entire pathways that are basically being shut down because you don't have a particular enzyme, or maybe the enzyme is not super functional, and so that would basically shut this pathway down, and then you have favoring of a different pathway instead. And again, that may be okay. You may not notice a whole lot of that, and depending on the disease state, or sometimes it could be fatal. Just depends on kind of what happens. So some examples of these disease states that may come about. So if you guys have ever heard of fetal ketonuria, what can't they have? That's kind of a popular thing. Yeah, fake sugars, right? So you can have like diet sodas, right? Because basically they cannot process those molecules, and so you can also see some other issues that pop up with those patients. Like epilepsy is a common thing you'll find with a lot of these. Usually some sort of developmental delays, like mental retardation, things like that can come about from these. I won't ask you to memorize all of these disease states. I'm just really kind of showing you as an illustrative purpose of like the different disease states that can come about from these different inborn errors of metabolism here. But it can happen with either amino acid metabolism. It can happen with carbohydrate metabolism, lipid metabolism. Anything under the sun could be affected by this, and sometimes it's just a single mutation that's causing these changes for these patients. You have huge effects on their quality of life. A lot of times these patients are so profoundly devastated by these changes that some of them end up being on a ventilator for the short period of time that they are alive. There's all kinds of different things that can happen to them, so it's pretty significant when you see it. Okay. There's actually an interesting one here. There's a glucose 6 phosphatase deficiency. So if you hear like G6PD deficiencies, that's a really important one for drug metabolism in a lot of cases. So you can have patients who will actually end up developing a methemoglobinemia, so where they turn blue essentially from that. So lots of different things that can occur here. Sometimes they'll be affecting drug therapy, sometimes they won't. Just depends on the mutation. Okay. So energy production, right? So you have to follow the laws of thermodynamics. So kind of going back to our basic physics here. So again, what's kind of the first law of thermodynamics? Like if you had to describe it to somebody. Never created nor destroyed. It just changes form, right? So and again, when we are producing energy, are we 100% efficient? Absolutely not, right? So we're always going to have some inefficiencies built into the system, and so we're going to look at that, why that is, and we're always going to have waste products. And then the other way that a lot of energy gets wasted in the body is by what? Heat, right? So again, we see this in certain disease states. You can actually find the body starts producing a ton of heat when it's having kind of poor energy metabolism occurring there. Have you guys ever heard of malignant hyperthermia? It's a disease state that can occur in sometimes patients undergoing surgery due to some of the general anesthetics we'll use for those patients. Sometimes there's another drug that can do it as well, but basically the muscles just start to get super tense. They basically are just in a constant state of contraction. When you're doing that, you're burning through a lot of energy, and that also produces a lot of acids and produces a lot of heat, and that can actually be rapidly fatal to those patients, and unless you kind of catch it early. But heat is going to be the major waste product that we're going to find through this metabolism of different products and how we get energy. And our main energy source of the body is going to be what molecule? Yep, so ATP, so adenosine triphosphate is going to be the major thing there, and we'll see that there's quite a bit of energy included in those phosphate bonds as we kind of go forward. But again, we're not going to be 100% efficient unless you're some sort of crazy robot or something, but again, you have poop at the end of the day, so again, we're not going to be 100% efficient. Stuff has to waste it, all kinds of different products. So also carbon dioxide, water, all kinds of different waste products. Again, again, we get all of our energy from the sun at some point, right? So again, if we're having that turn into, eventual, turn into plants. Plants, get eaten by animals, and we eat the animals. It's a circle of life, basically, right? Hold your tiny lion up to the sky. I tried doing that with my daughter after she was born, and the nurses did not appreciate that. Be careful. So anyway, so again, we mentioned that we're not 100% efficient, so there's going to be some energy that's lost as we go through these different steps. It's just kind of this law of entropy that's happening here. But as we go along, we're going to see this free energy is going to be decreasing as we go through from step to step. So for instance, if we're just taking in food into our digestive tract, we're going to be losing some energy lost in that feces, right? Some amount of usable product might still be there, and again, you can use that for other things, like maybe use it for fertilizer or something. But then you have your digestible energy. Some energy is going to be lost either through the urine or different gases and things like that. And then we actually metabolize it. That's where we're going to see heat actually starting to be produced there. And then finally, when you get down to the end of it, we get this kind of net energy that we're developing. And that's usually going to be in the form of ATP, which is our energy currency within the body. Again, just another idea here. If you see just constant energy is going to be dropped from step to step to step, and eventually usually the end products we're going to be seeing with metabolism, especially with like glucose and things like that, are going to be carbon dioxide and water, right? So that's especially when we're undergoing aerobic metabolism, and aerobic metabolism requires what? Yeah, so we're going to see, especially with oxygen being utilized for oxidative phosphorylation, utilized in aerobic metabolism, you're going to get CO2 and water being formed kind of as the end there. More into detail in these steps as we go forward. We measure energy, at least in the food that we're intaking, as calories. You guys know where the term, like what an actual calorie is? How's that measured? Yep, exactly. Yeah, so there's kind of an archaic, way that they determined what a calorie was, and it was raising, 1 cubic centimeter of water, 1 degree Celsius, essentially. That's not super useful for our purposes, right? Now, normally when you talk about calories, like if you say, like, your candy bar has, 200 calories in it, we're actually talking about kcals, right? So you're actually standing for thousands of calories when you see that. So again, if you ever look at your candy bar, you're like, man, that's actually a lot of calories. 1,000 more than you actually thought. But we always just kind of refer to it as that. So if you ever see kcal, or we talk about calorie, at least in terms of medicine, that's just what we're referring to. Basically using the term synonymous, even though it's probably not quite right. Again, just an example of seeing how energy can be, obviously, what kind of molecules do we get kind of the most energy from? If you're looking at the number of calories per gram of a substance. Usually fats. You know how many calories come in a gram of fat? Yeah, roughly 9 calories per gram of fat. How about for carbohydrates? Actually 4. Yep. So you get less energy. But what's kind of the difference in the ability to utilize that energy between fats and carbohydrates? Which one's faster? Yeah, carbs are a lot faster, right? And we'll see why that is in just a little bit. But yeah, so you can utilize those carbs a whole lot faster in a lot of cases. But for less energy, though, on an actual gram per gram basis. And then how about proteins? 7. Actually, proteins is going to be 4 again, yeah. So, and we utilize those calculations quite frequently when we have to figure out nutritional demands for our patients, right? And so if you had a patient, and I do this most frequently for our patients when we have to do total parenteral nutrition. Do you guys know what that is? Can anyone describe what it is? Yeah, sometimes we'll utilize feeding tubes. We talk about total parenteral nutrition. That's mainly referring back to IV nutrition. So we're actually giving all your lipids, proteins, and carbohydrates, and all the other minerals and nutrients right through the IV line, right? So that's where pharmacists get involved, mainly because a lot of those products are handled as drugs. And so we have to figure out how much energy the patient needs per day in terms of like how many calories they require, and that can be dependent on what kind of disease states they have. It can be dependent on what their current weight is, what their age is, different things like that. And then we figure out, like, okay, well, we know how many calories they need. Now we need to figure out how many grams of protein, carbs, and lipids they need in order to satisfy that in an appropriate amount. We can't just give them all fat, right? Because that would lead to all kinds of problems. We need to give them some carbohydrates, some fat, some proteins in order to keep a balanced nutrition set, right? Anyway, you can see some comparisons there. Obviously for 100 calories, you get a whole lot more carrots than you do potato chips. Obviously, you guys it's pretty intuitive to you guys, I'm sure. Right, so as we break food down, the energy has to be converted into that usable form, and so we'll talk about those steps in just a little bit, but eventually we're going to get our energy currency, which is going to be ATP, and it's going to be our universal carrier for energy throughout the body. And ATP is formed where? Right, that's why they're the powerhouse of the cells, right? Because they're forming our ATP in a lot of cases. So we do have some hydrogen carrier molecules, and these are really important for carrying hydrogen molecules and these protons through the processes we're going to see in a little bit in order to eventually get them to oxidative phosphorylation. That's going to be that final step that we have in order to actually produce ATP in the greatest efficiency that we can, right? And that's going to be where we're going to require the oxygen in order for that to occur. So you have NAD. You can say nicotinamide adenine dinucleotide, we'll just call it NAD for now. That is able to accept a proton, or accept a hydrogen proton, and carry it through the different steps of energy production. This is actually derived from niacin, or vitamin B3. You guys ever taken niacin before? What do you normally take niacin for? You have a heart disease, right? It actually helps with cholesterol, right? Do you guys know what happens when you take niacin, what it does to your body? Yeah, you get really flushed. I actually had some friends in pharmacy school who decided to do a niacin challenge. We have delayed release formulations now that don't really lead to so much flushing, but they took a whole bunch of immediate release niacin, and then I was actually picking one of them up from their rotation, and she gets into the car and just beet red. She'd been vomiting the last hour just because of how much histamine was released in her body, and just all this miserable for like the rest of the day. So be careful with immediate release niacin, no fun. But it's very useful because it helps to produce NAD, and that's going to be good for helping us to produce ATP. FAD, or this flavin adenine dinucleotide, comes from riboflavin or vitamin B2, and so that is going to be able to accept 2 hydrogen protons and be able to carry those again to the mitochondria for energy production. These are considered coenzymes, and they facilitate this hydrogen transfer. And we'll see as we go through glycolysis and the Krebs cycle and all of that where those come into play. I've never thought I'd have to go through the Krebs cycle again, but guess what? Krebs cycle's coming. Okay, so that concludes the second lecture. Any questions on that section? Or continue on. Yes, ma'am.
You said NAD can accept 1 hydrogen?
Mm-hmm. Yeah, and then FAD will accept 2. Yep. Continue on. Lecture 3, so we're going to get a little bit more into the nitty-gritty of energy production and cellular metabolism, and then we'll also talk about the extracellular environment. Okay, so we should be able to discuss, again, just kind of going through our basics of organization and function of cells, and then we'll also discuss membrane physiology and action potentials, and that's going to be really, really important towards the end when we're talking about electrolyte balance between the outside and the inside of a cell, how we're able to conduct action potentials. That's really important for not only the nervous system, but also for the heart. As we'll get into more detail, we'll talk about the cardiovascular system in the second section of this class. So anywho, so let's go and just get into processes here. Without an adequate supply of blood and blood carrying mainly oxygen to our tissues, we get ischemia that's happening. How would you describe ischemia to somebody? Lack of blood flow, okay. And what happens when you have lack of blood flow? Oxygen deprivation, then what happens?
Cell death.
Yeah, you can see cell death, basically. So again, if you have ischemia to the tissue, eventually the cell dies because it doesn't have any oxygen, right? Think about how you would describe this to someone who didn't know anything, because again, you'll find a lot of patients who don't know anything, right? And so if they ask you, like, what is myocardial ischemia? Like, what does that mean? You can describe it to them, right? But anyway, another term we may say is hypoxia, but obviously, what's the main carrier molecule of oxygen within the body? Yeah, so hemoglobin, we'll get into more detail on that later on in the class, but that's going to be our main thing, is carrying oxygen to the tissues. And again, when we don't have oxygen available to the tissues, what do we do instead to produce energy? Yeah, so we'll do anaerobic metabolism. We'll see how we can look between those two there, and how which one do you think is more efficient? Yeah, aerobic is always going to be more efficient. Again, anaerobic is good for kind of short-term use of energy, so especially like when you're exercising or something like that, that's going to where that is most prominent. So anywho, so a couple of different metabolic processes we're going to be discussing here. First one's going to be glycolysis. That's basically we're going to be converting glucose into 2 molecules of pyruvic acid. This is going to be the first step towards oxidative phosphorylation we'll get into. We'll have glycogenesis. Glycogenesis is going to be what, do you think? Yep, so it's going to be production of glycogen, and glycogen is what? What does that mean to us? Yeah, so it's going to be energy storage, glucose storage. Primarily, where do you see glycogen stored at? Liver's a big place. Where else do you see small amount? Muscle's a good one. Yeah, like the heart tissue can have a small amount. And again, that's we'll see that depletes pretty quickly, especially in the muscle when we have short bouts of anaerobic metabolism going on there. Next, we have glycogenolysis, and that's where we're going to have that breakdown of glycogen into free sugar that we can put back into the bloodstream. That's where the liver is going to be really important there for glycogenolysis. Do you know what type of hormone is going to influence that? We've talked about already. Yeah, glucagon is going to be the big thing there, right? So you're sensing you don't have enough blood sugar in the bloodstream. It's going to go ahead and send out, your pancreas will send out glucagon, and that'll trigger glycogenolysis, right? Breaking down that glycogen to produce new glucose. And then following glycogenolysis, we're going to have gluconeogenesis, which is just that new production of sugar. We'll look at those kind of the interplay between those two steps in a little bit. Next, we'll have lipogenesis, and that's going to be the production of triglycerides, primarily going to be in the adipose tissue, right? So if you have too much energy floating around, too much blood sugar, eventually that can get stored as fatty acids, and that's where we're going to see lipogenesis. And then the opposite would be lipolysis, breakdown of the fatty tissue. And again, we can utilize fat for energy. It's just not going to be as a quicker process as we will see with glucose. And then finally, ketogenesis is going to be that state where we're kind of depriving our cells of glucose for a long enough period of time. We have to start resorting to metabolizing other products like proteins, like fats, and that's where you're going to see a lot of free fatty acids are being formed there, and a lot of ketone bodies, and that can lead to things that we mentioned, especially with like a type 1 diabetic, lead to what? Yeah, DKA or diabetic ketoacidosis, right? So a little bit of ketosis, probably okay. Too much is going to be a problem, right? And so we mentioned that sometimes we'll actually put patients into ketosis on purpose. I mentioned especially for patients that have really bad intractable seizures. Sometimes we like to acidify them a little bit. That makes it harder for them to seize. So we'll put them on a ketogenic diet. Obviously, you guys are familiar probably with the diet, not fat, I should say, but the diet, like a keto diet, where basically you're intaking very low amounts of carbohydrates in order to induce a degree of ketosis, because that's where you're breaking down a lot of fats and things like that. So again, basically the two terms here, we have catabolism, which is going to be the breakdown of molecules. Usually that's going to help us free energy as we do that. And then you're going to have anabolism, as we're going to be putting those back together to use some sort of larger molecule. Usually that requires energy. Okay. All right, so as far as catabolism goes, we're going to see that we can break down lots of different products here in order to produce energy. So we can break down amino acids from proteins, we can break down fatty acids from our fats, and we can break down glucose or other types of carbohydrates in order to get energy, right? And so you can see here where we have these energy production. Eventually, we're going to have some degree of oxidation that happens here. We'll have ATP that's being produced within the mitochondria. So you can see this step happening here, where eventually either your fats or your amino acids get converted over to acetoacetic acid. Right? That's also where a lot of our ketones are coming from. Glucose itself gets converted to pyruvic acid. That's where that glycolysis pathway comes from. But eventually, it's going to turn into ATP. Right? It's our main energy production there. Also notice you're going to have CO2 being formed here and water. It's going to be our major byproducts of the oxidative phosphorylation there. But basically, we're going to have ATP that can be utilized for energy. So we can see it being used for things like active ion transport. We'll talk about different kind of transport proteins within the cell membrane that utilize ATP as a means of energy, as a means of actually doing its process. We'll look at why that works in just a little bit later. But utilize it for cell division, synthesis of new products, all kinds of things you use ATP for. And then you're going to get ADP, which is just adenine diphosphate plus a free phosphate molecule. And that can be later used to produce ATP again later on down the line. Right? So we can keep recycling our ADP into utilizing the ADP once again. Now note here that the phosphate bond that occurs with ATP, and we notice that there's how many phosphate groups on the ATP? 3, right? It's right in the name. When you break one of those down, when you cleave that phosphate off, you release a large amount of energy. And roughly for 1 mole of ATP, you guys remember how many molecules are in a mole? Yes, yeah. You go back to not Avocado's number, but Avogadro's number, right? 6.022 times 10 to the 23rd. Basically, if you had that many molecules of ATP, if you broke down that phosphate bond for every one of them, you eventually get 12 calories, right? Or 12,000 calories in the strict sense, but 12 kcals essentially. So a decent amount of energy that we're actually getting from that amount of ATP there. And you can see what the actual structure looks like here. Again, the same adenine you would find in your DNA or your RNA, the same molecules seen here. Okay, so this is occurring in 3 different steps. The first one's going to be glycolysis. It's going to be occurring within the cytoplasm and does not require any oxygen. So this is an anaerobic process that we're seeing there with glycolysis. Okay. Next, we're going to see the Krebs cycle. Again, this is going to be require some amount of oxygen, so this will be an aerobic process, and this is going to be occurring within the mitochondria. And then finally, where we get the main majority of energy that we're going to be developing, or main amount of ATP, is going to be in the electron transport chain, and that's occurring directly within the mitochondria. This is going to be absolutely an aerobic process, requires a lot of oxygen, produces a lot of carbon dioxide in order to get those ATP out at the end. We'll see that if you don't have any oxygen around, you switch over to anaerobic metabolism. That's where we're going to end up producing a lot of lactate, right? So again, why do you feel sore after you work out? You have a lot of buildup of lactic acid, so it takes a little bit of time for your body to actually clear a lot of those. There's also other reasons why you're sore as well, but lactic acid is usually one of the common reasons why you think about muscle soreness. So anyway, so again, we either are getting energy, and specifically we're talking about glucose here for these steps, either glucose coming in from the digestive tract, right, so just for the food we're intaking, or it's being broken down from glycogen, say within the liver, undergoing that glycogenolysis and then gluconeogenesis, and releasing glucose into the bloodstream. Okay, once it's able to get into the cytoplasm, and can glucose just kind of freely diffuse into the cytoplasm? What does it require? Some sort of carrier molecule, right? But what usually triggers that? Yeah, insulin can be a big trigger for that in a lot of cases. So insulin is going to be one of the primary hormones that helps to influence glucose uptake into the actual cells, because you want your cells to be impermeable to glucose in most cases. Usually too big of a molecule to actually cross that lipid bilayer, but we're going to need some sort of way to get it in. It's usually those carrier molecules. So anywho, once glucose gets into the cytoplasm, this is where we're going to have glycolysis occur, and then we're going to get pyruvic acid formed. We'll go into more detail on how that occurs. And then at this step, you can either undergo aerobic metabolism. We're going to go through the Krebs cycle, the citric acid cycle, and then electron transport, which is that oxidative phosphorylation. That's going to be the aerobic pathway, right? That's going to be the most efficient, produce the most amount of ATP. Otherwise, you can see the pyruvic acid being utilized for anaerobic metabolism, in which case the byproduct is going to be lactic acid. We'll talk about this as being the Cori cycle. We'll go into that more detail on that a little bit later after we get done talking about that aerobic metabolism. Everyone's still with me so far? It's only going to get rougher from here. Buckle up. Okay, so basically what's happening with glycolysis, and again I had to review all of this because I hadn't had to cover it since biochem in farm school. So again, this is, if I say something wrong, please call me out for it if you guys have had it more recently. Anywho, when you have glycolysis, basically we're taking a molecule of glucose and we're breaking that down into 2 pyruvic acid molecules. Okay, so 1 glucose turns into 2 pyruvic acids. It'll be important. I'm not going to ask you to memorize every single step along this pathway. That would be very mean, and I had to know stuff like that when I was in farm school. But you should know what the end products of these different steps are going to be, right? So you need to know, like, okay, where's ATP being produced at, how much ATP is being produced, and where's like our hydrogen carrier molecules coming into play, and all those things like that. So we'll go over that in more detail, and I'll kind of show you kind of the summative steps as we go along of kind of where we're at in the process. The first step here, glycolysis, breaking down glucose into 2 pyruvate molecules. Notice here, this actually does require some degree of energy for it to occur. So you end up losing 2 molecules of ATP in this process here, but towards the end, you actually end up developing some hydrogen molecules, right? So you're going to get some protons here that can later be used for oxidative phosphorylation. You're also going to have 4 molecules of ATP that are being produced here as well. Okay, so we're utilizing 2 ATP, but we're also producing 4 during this step. So the net gain in ATP out of this step is going to be 2 molecules of ATP, right? So again, you're thinking like, well, 1 glucose makes 2 ATP. That's not very good. It's okay. It's going to get better. We're going to get better yields on that later on as we go through the citric acid cycle and oxidative phosphorylation. Okay, so again, the only net energy gain we have is going to be this ATP, even though we produce 4, 2 end up getting utilized in those kind of first steps. And again, notice everything after this pathway, when we talk about pyruvic acid going into the citric acid cycle, that's happening twice for every glucose molecule that we had to start out with. Okay, so again, everything we'll see you're going to be doubling it essentially because it's going to be happening twice based on that. And again, glucose, you can see here, the carbons are just basically being cleaved in half. Glycolysis is happening here, and you get 2 pyruvic acid molecules at the end. Notice we're also getting these the NADH, 2 molecules of that. That's where we're going to have most of our protons being kind of hoarded until we get down to the mitochondria for the oxidative phosphorylation. Okay, and again, if you were looking at kind of the free energy of this system and how that goes along, again, I'm going to have a lot of pictures that show kind of the same thing, just in different ways, just to try to cement it in your minds. But the energy is going to go up as we utilize the high energy phosphate bonds here and 2 molecules of ATP. So kind of here we're at the highest energy state, and then we're going to end up utilizing NAD in order to hold on to a couple hydrogen protons, and then also generating a few molecules of ATP as well, right? And so we can utilize these later on in the process. Again, just our net products we have here, 2 pyruvic acid molecules, 2 NADH, plus 2 kind of free hydrogen ions here, and then a net gain of 2 ATP. I won't try to trick you on the exam. I won't be like, well, how many, if I say how many, ATP molecules are net gained from glycolysis, you guys will know it would be 2, right? I'm not going to try to trick you up on the test for this step in particular. You might think I'm trying to trick you up for other things, but it won't be on this one at least. Okay, and again, the anaerobic pathway kind of ends at this state, right? So once you have those pyruvic acid molecules, you're actually going to see that if you don't have any oxygen around, you can't really do the Krebs cycle, can't do oxidative phosphorylation. We can utilize this ATP at least for some amount of energy, and then we're going to find that we can store that pyruvic acid in a different form for use later, right? That lactic acid is not going to go to waste in the long term. But essentially, and we'll talk more about this in detail a little bit later, but this is kind of the beginning of that Krebs cycle, where essentially, if you don't have any oxygen here to complete the breakdown of glucose, you're going to have lactic dehydrogenase. That's going to be an enzyme that's actually going to be able to convert that pyruvic acid over to lactic acid, which can then be later utilized. It can be backwards converted over into glucose eventually, as you'll see. It's not going to be as energy efficient as if you could have used oxygen in the first place, but it is a way we can actually utilize that lactic acid later on, and that's primarily going to be occurring within the liver. So, say for instance, like you're working out, you go into a state of anaerobic metabolism in the muscles. It's going to generate a lot of lactic acid that eventually is going to get carried in the bloodstream back to the liver, where it can be recycled. Okay, we'll look at that process later on. But basically, by doing that, you notice this is going to be kind of a process that can go kind of either way. By converting the pyruvic acid over to lactic acid, you end up kind of freeing this side of the equation, and you're allowed for glycolysis to continue occurring, right? Because again, if you had too much pyruvic acid build up, that would eventually kind of shut down that feedback inhibition and prevent further glycolysis. But because we are kind of shuttling it over into lactic acid, it can be eventually given away to the liver, and we can have further glycolysis occurring. So not super energy efficient, only get a few ATP out of that, but it's better than nothing, right? Okay, so we're going to go back to our aerobic pathway because we're much more interested in, much more efficient. Okay, so here, once we have our pyruvic acid, that's going to be entering the mitochondria, where it can then be used in the Krebs cycle, otherwise known as the citric acid cycle. So pyruvic acid leaves that cytoplasm, is going to enter into the mitochondria here. So again, this is where this is taking place. Eventually, we're going to be removing carbon dioxide to form this acetic acid. Acetic acid is then going to form with acetyl-CoA or acetyl-CoA, and eventually we're going to end up utilizing, acetic acid formed with coenzyme A to form acetyl-CoA. That's kind of our end product here, and that's what we can utilize in the Krebs cycle. Okay, so before you get to the Krebs cycle, pyruvic acid gets converted into acetyl-CoA. Okay. Again, this step here, there's no ATP that's actually being formed here. However, those hydrogen ions can be oxidized later and be utilized for ATP. Okay, so we'll utilize those later once we get down to the oxidative phosphorylation step. Okay. Again, just to show you what this looks like, pyruvic acid being combined with this coenzyme A is going to get converted over. And you notice here, there's a 2 here, right? Because again, for every glucose molecule, we're going to get 2 pyruvic acids. That gets converted into 2 molecules of acetyl-CoA plus some carbon dioxide, because we have to get rid of those carboxylic acid groups. And then we also get those 4 protons. Okay. Okay. When I was in school, I had to memorize every step of this process. Did anyone else have to do that? Okay. I actually got a tattoo because I was just like, I'm tired of having to memorize this. I have to use a series of mirrors to see it, but don't worry about it. Anywho, I'm not going to make you guys memorize every step of this process, but again, I want you to know kind of what the input is going to be into the system and then what we're getting out of it, right? That's kind of the important thing to note from this. This is occurring within the matrix of the mitochondria, so occurring right within there. And eventually that acetyl-CoA gets converted over to citric acid, and then that citric acid is going to be kind of the start of the process here, right? Notice this is a cyclical process. We're going to be able to continue this on as long as we have substrate to enter into the cycle. And notice what we're going to be producing here. Some small amount of ATP, but the main thing we're doing is getting a lot of protons, getting ready for the rest of the mitochondria to utilize an oxidative phosphorylation. Essentially, one pyruvic acid molecule. Again, you should double this when we're talking about one glucose molecule. ADP is going to form 1 molecule of ATP. You're going to find that NAD is going to accept 3 hydrogens. So you're going to get 3 molecules of NADH plus 3 free hydrogen ions. And then you're going to have 1 molecule of FAD turned into FADH2 because it's accepting 2 hydrogens there, right? So basically just accepting hydrogens, holding on to them until we can utilize them later on. Okay, we're also generating some carbon dioxide here as well. So again, this is a aerobic process, requires oxygen in order to form that, in order to continue on, and we get CO2 as a byproduct. So again, pyruvate coming in here from glycolysis, you're going to get some NADH being formed. I'm sorry, this is where it gets formed with the CoA to get acetyl-CoA. That enters into the cycle. You're going to lose some carbon dioxide, get some NADH, get some ATP out of this, some FADH2, and then we can get ready for the last step. Okay, I'll have some slides where I actually kind of summarize where we're getting all of our protons from in just a second. Again, just another a little bit more condensed view of kind of what's happening here. But again, at the end of the day, what we're getting is 3 molecules of NADH plus 3 hydrogen ions. So it's going to be 6 total hydrogen ions there. 2 from the FADH2, right? And then 1 ATP and 2 carbon dioxide. All right. Here's the more detailed step if you were ever curious, if you wanted to like draw it out yourself for fun or anything like that. Go wild. Just looking at that brings back nightmares. So, yeah, probably for you guys too. So anyway, so where we're at so far in the process. So 1 glucose, we've gone through glycolysis. We get 2 pyruvic acid molecules, and basically what we see with each of these, when we add them together, is we have now 6 molecules of NADH. We have 6 free protons here. We're going to have 2 molecules of FADH2. 2 ATP that have been formed, kind of as a net gain here, and then 4 molecules of CO2. So this is just through the step of the Krebs cycle, right? So it did not include the byproducts of glycolysis, although we'll add those up later on in a few steps. So everyone with me so far? Okay. So basically glycolysis, we have 2 pyruvic acids, we have 2 ATP, and 4 hydrogen ions that are being produced here. Through conversion to acetyl-CoA, this is where we're also going to get basically a pyruvic acid turns into acetyl-CoA. We're going to get 2 molecules of CO2 and then 4 hydrogen ions. And then through the citric acid cycle, this is where we get the main source of our hydrogen ions. We're going to get a net of 16 hydrogen ions here. Again, a lot of those are being held up by NADH or FADH2. We're going to get some degree of 2 ATP here. And so as a total, now we have, going into the citric acid cycle, we're going to have 4 ATP and 24 hydrogen ions. Okay. So now we end up going to our last step, which is, everyone's been waiting for it, electron transport chain, oxidative phosphorylation. Essentially, this is where 90% of the ATP production actually happens here. So you're getting these hydrogen atoms are being split into a hydrogen ion and an electron. So that splitting that happens there. Eventually, you're going to end up getting water that's being formed out of this process, and you're also getting a CO2 that happens there. But essentially, and again, there's lots of different kind of coenzymes that are being utilized here to generate this ATP. But what we're going to see is that basically for every 2 electrons that pass through, we end up getting 3 ATP generated from that. Okay, so 2 hydrogen ions, when it goes through the electron transport chain, ends up generating 3 ATP molecules. So for the 20 hydrogen molecules that go through this, we're going to get 30 ATP out of it. Okay. Pretty efficient from that standpoint. We're going to get an additional 4 hydrogen atoms, end up making 4 ATP on their own, right? So they don't go through the full chain. So as a net utilization of 1 molecule of glucose, after all is said and done, we basically get 38 molecules of ATP. Okay, and so if you were to actually go back and look at the possible energy that you could get if you had 100% conversion efficiency, you know, we see that we're not that good. We're about 66%, right? And I think if you guys got a 66 on the test, you'd be pretty bummed, right? It's a failing grade. But for our bodies, it's good enough, right? So we say, you know what, C equals farm D, as we used to say in farm school at least. But the remaining 34% of it, I was just kidding, I made very good grades. The remaining 34% ends up getting lost as heat, right? Because again, we cannot, we can only change form of energy. We're not making anything, we're not losing anything. The rest of it just gets lost as heat, right? What's interesting here, at least as far as clinical corollaries, I know I've been relatively light on those, going through the kind of biochem processes. This cytochrome A3, this is actually a really, really important enzyme in the tox world because this is actually where cyanide works. So cyanide actually, you know, if you ever, you know, when you think about cyanide, what do you think about? That poison, right? Like someone like trying to murder you, or like, you know, if my wife wanted to, like, if I made a bad comment about her being pregnant, like, you know, her trying to murder me, she might go through cyanide, right? Does anyone know what cyanide smells like? Yeah, it just smells like almonds. Yeah. Basically, cyanide actually comes in and actually will inhibit this enzyme, cytochrome oxidase A3, and prevents the utilization of oxygen. So even though your body is transporting lots of oxygen through hemoglobin to the tissues, it can't utilize it. So now all your tissues they're starved for oxygen. They go anaerobic, and then you end up generating all these different acids, lactic acid being the primary one. You get very, very acidotic, you get very, very hypotensive, and eventually they die, right? Basically because you're just starving the tissues of oxygen. So it's kind of an interesting way you can utilize you know, certain molecules to interrupt the system for better or for worse. Does anyone know what type of foods contain cyanide?
Castor beans?
Okay, you actually get ricin from castor beans. Oh yeah, what part of a cherry? The pit, yeah, actually. So you think about like cherry pits, think about peach pits, apple seeds, actually. If you were to chew all those up, there's actually a product in there that gets converted over into cyanide. So if you ever have someone says like, oh no, I swallowed an apple seed, they're okay, as long as they didn't chew it up, right? And it takes a lot of apple seeds to be a problem. When I was at the Poison Center, actually, I had a guy call up and said, I just ate a peach pit, and I read online that it has cyanide in it, am I going to be okay? And I was like, did you actually chew it up? And he's like, yeah, I chewed it up. I'm like, who are you? Like, how do you have a jaw that strong? I was much more interested in the actual mechanics of him chewing this thing than his health at that point. He did fine though. He was fine. He did not get near enough cyanide to actually be a problem. But interesting enough, there's actually one drug product we'll actually use that actually contains cyanide as well. It's a drug called nitroprusside. It's actually one we use to lower blood pressure. It's an IV medication we'll use like in the ICUs that actually has molecules of cyanide contained in it. That if you get too much of it, can actually lead to a problem similar to an actual poisoning. So we'll talk about that more when we get to the actual pharmacology lectures. So. Correlations there. Anywho, so we see that we're not 100% efficient with our ATP production, but we're pretty good, and our bodies are okay with that. Now, as far as storage goes, we don't need to undergo a lot of glycolysis. We're not really needing to produce a lot of energy. This is where glycogenesis occurs here. And notice this is going to be a two-way street, right? So pairing up glycogenesis with glycogenolysis, either process can be occurring based on what our needs are, right? So for instance, insulin would stimulate which process, do you think? Glycogenesis, right? Because we said that when your blood sugar is too low, we need to utilize those glycogen stores, and that's where things like glucagon are going to come along and try to stimulate breakdown of that glycogen. On the other hand, insulin, because we're in a state where we're trying to store up energy, that can lead to this glycogenesis actually occurring here, right? And it's all dependent on what your blood sugar is doing and how your body's responding to that. We'll get more into that in the endocrine section. But essentially, you're going to find that glycogen is going to be mainly stored within the liver, but you can also have some small amount being stored in the skeletal muscle tissue, which is good when we're undergoing like an acute bout of exercise or something, and then cardiac muscles as well, right? We like to have a little bit of energy storage for our cardiac tissue. Otherwise, if it doesn't have any energy, you don't have any energy and you're dead, basically, right? Note that this is a process where blood glucose is getting taken in. Say this is a hepatocyte. You're going to have this hexokinase that actually utilizes some degree of energy here. Right? Utilizes one molecule of ATP to get this glucose 6-phosphate. Okay. That can then later be stored through a series of different enzymes, and eventually you're going to end up getting this glycogen actually occurring here. You guys remember what the kind of the structure of glycogen was? It looks like a big chain of sugar molecules stuck together, essentially, right? And then when you have something like glucagon come along and say, hey, we need to stimulate blood sugar, you're going to have that come along, try this process of glycogenolysis. We're actually going to be removing that phosphate group eventually. You don't regenerate that ATP, so notice that we're not really getting as much efficiency as if we use the glucose itself without going through this step. But again, this allows us to store it efficiently for our bodies for later use, right? So it's okay to invest a little bit of energy into that process. So we already kind of mentioned this. Basically, end up yielding free glucose to be secreted into the blood. Note that the skeletal muscle tissue and the cardiac tissue, they want to keep that glycogen for themselves, so they're not really going to be spilling that out into the bloodstream. Really just the liver that's going to be the primary one that will release glucose back into the bloodstream after it's been converted from glycogen back to glucose. And we mentioned that things like glucagon in the pancreas can stimulate this process, but also things like epinephrine. And if you think about like your sympathetic nervous system, like if you had a fight or flight response, a bear hopped out at me, my adrenal glands are going to start to pump out a lot of epinephrine. I need glucose, I need energy to run away. So that's going to go ahead and be responsible for also stimulating glycogenolysis. So again, kind of just similar to what we were already saying here. Notice that the liver has that enzyme glucose 6-phosphatase. That's going to be removing that phosphate group, and that's what allows us to kind of get back to the baseline sugar molecule to be released into the bloodstream where we can utilize that in other types of tissues, right? But important to know that the heart and the skeletal muscle tissue is not going to be releasing that glycogen. At any point they want to use it. They're very selfish. They want to use it for themselves. The liver is kind of the giver of the, kind of rhymes too, of the body. Okay. So this Krebs cycle, this is where we kind of get back to our anaerobic metabolism and how we can utilize that lactic acid that gets produced during that process. So again, during exercise, we can't supply our tissues with enough oxygen, so you end up undergoing that anaerobic metabolism there. So the main byproduct is lactic acid, as we mentioned. So lactic acid is eventually going to get transported out of the skeletal muscle tissue into the liver, and there it can be worked on by that lactic acid dehydrogenase enzyme. Right, so again, we saw that the arrow went both ways on that pathway. You can convert the lactic acid back into pyruvic acid. Okay, so basically when that occurs, you're going to get pyruvic acid, and then now you have NADH plus another hydrogen ion, and then that can go into the process can go into the Krebs cycle and go into oxidative phosphorylation. It's able to be utilized. Right, so you can't utilize lactic acid immediately, but at least it's a good storage molecule for that pyruvic acid. Again, here in the picture, you see that something like epinephrine stimulates breakdown of glycogen within the skeletal muscle tissue. Say if you have a fight or flight response, you end up getting that glucose 6-phosphate. That eventually will end up being formed back into, when you undergo glycolysis, into pyruvic acid. If you don't have enough oxygen around to undergo the TCA cycle and the oxidative phosphorylation, that's where you get lactic acid, right? So this is an anaerobic process. Once it gets shuttled over into the liver, there it can be converted back over to pyruvic acid through that lactic acid dehydrogenase, the LDH, and then that can be converted back over to eventually glucose, or it can be stored as glycogen, whatever your body needs at that moment. Okay, so it's just a process for us to kind of recycle that lactic acid, so that way it's not a total waste. We still can get a decent amount of energy out of that. So again, just another picture kind of showing the same thing that's happening here. Note that this is an ATP dependent process, so we end up losing some energy by undergoing this pathway. You end up losing about 4 ATP, so you can't do this forever, right? Your body can't subsist on just lactic acid alone, but eventually, it's good for kind of short-term use, especially after a sympathetic response. And eventually you can actually overwhelm the body in a lot of cases in developing a lactic acidosis, which can be very detrimental to the body. So there's cases like if you have sepsis, or if you have a really bad bloodstream infection, you're getting hypotensive, this can lead to a lot of lactic acid buildup, which can be problematic. We see this with certain drugs can actually cause lactic acidosis. So again, it's not a state we want to be in, but it's okay in kind of short-term short-term situations. Okay, and again, just another picture kind of showing the same process here, just to give you different ways to kind of look at the same thing. Okay. So any questions on that so far? Okay, so that's the Krebs cycle. See what we end up producing there. So again, turning lactic acid into pyruvic acid. We mentioned that was mainly just metabolism of carbohydrates, right? But we mentioned that you can utilize energy from other sources as well, mainly our amino acids and also from our fatty acids as well, right? So lipids, proteins, they can also be used. It just takes, it's not nearly as efficient as it would be if we were using just glucose itself. But again, if you're in a pinch, if you don't have enough blood sugar, that's when your body kind of switches over to utilizing those kind of other sources there. So eventually, you're going to find that all these different products will eventually turn into acetyl-CoA, right? That acetyl-CoA is going to be that initial step for the Krebs cycle. So that's that pathway downstream, but that is all going to be the same as it would be for your glucose. But actually, with the process happening here, I'm trying to see. Right. So for instance, if you see your glucose molecule coming into this state, this is where it undergoes the glycolysis into the pyruvic acid. On your other hand, if you have something like a fat molecule, like a triglyceride, right? You guys remember the structure of the triglyceride? You had like glycerol molecule, and then you had 3 fatty acid chains coming off of that. Well, all those carbons can be utilized for energy production. So basically, the fatty acids themselves can be converted into acetyl-CoA. You can also have that glycerol being utilized in this process as well. So glycerol itself can be turned into pyruvic acid, because remember, glycerol is just 3 carbons long, so very similar in structure to that pyruvic acid. So kind of using all parts of the animal here, essentially to generate energy. And again, that's why we get so many more calories per gram of fat than you do like a carbohydrate, just because there's so much more kind of stored up in those bonds already. Okay, so lipogenesis, that's going to be our process for actually generating new lipids within the body. Again, when the glucose itself doesn't complete glycolysis, so again, say for instance you were to have too much blood sugar in your system, you don't really need all that extra energy, right? So because that glucose can't undergo glycolysis, it needs to be stored somewhere, right? And so this is where you can actually have fatty acids that are being formed. So essentially you're taking those sugar molecules and attaching them onto this glycerol, and now all of a sudden you have a new triglyceride, right? So that's why if you have too many carbohydrates, that's where it gets stored at, is eventually it's going to turn into fat, right? So even though you eat, all low-fat foods, already has kind of the fad back like in the 90s and stuff, realize that really wasn't the main problem. That's why we see a lot more low-carb diets nowadays, because we understand that having too many carbs in the system is what leads to a lot of fat production, and this is the process that's happening there. And then just on the flip side of that, we have this lipolysis. So we're going to have the hydrolysis of the triglycerides. Basically, by freeing up those free fatty acids, you can utilize those carbons to go into the Krebs cycle eventually by turning it over into the acetyl-CoA. And so the enzyme lipase is going to be the main thing that's utilized for doing this. So lipase is able to cleave those bonds, and we can utilize those molecules for energy production. Okay, I think I'll give you guys a break now. We'll come back in 10 minutes and then continue on with our talk. Any questions for us? You guys go. Okay, I'll see you in 10 minutes. Questions from the first half? All right. So anyway, continuing on with our talk about energy production from other sources than just carbohydrates. So again, we were mentioning our adipose tissue. So that process of breaking down the fatty acids is going to be lipolysis. And again, we're going to get kind of two main things from that process. We're going to get, and again, lipase is the enzyme responsible for that. We get fatty acids. Those themselves can enter through the Krebs cycle by eventually getting converted over to acetyl-CoA. This is going to happen through this beta oxidation that happens here. So you can see the adipose tissue, when you have triglycerides that are being broken down to their resultant fatty acids, that can be transported within the bloodstream, right? So and eventually into the muscles. So if you get kind of into like a prolonged state of having, say, low carbs or not enough blood sugar, like this is where you can start to have this beta oxidation that occurs, and then you get acetyl-CoA. We'll talk about where ketosis happens downstream of this process. The glycerol itself, that 3-carbon molecule, is able to be transported, and within the liver actually gets converted by this glycerol kinase, and eventually is going to turn over it back into glucose. So again, kind of using all parts of the molecule to eventually get energy production. And then once this glucose can go and be utilized normally, as we've kind of mentioned previously. All right. So what happens is if we have too high a rate of lipolysis, especially if it exceeds kind of our fatty acid utilization, so we're kind of breaking down all these fat molecules, are getting way too many fatty acids floating around. So if you're in like starvation or like type 1 diabetes, if we have like, you kind of go too hardcore on your paleo diet or caveman diet, whatever, this is where the fatty acid levels are going to keep increasing within the bloodstream, right? And so the liver cells can only do so much. They're trying to convert the fatty acids over into acetyl-CoA through that beta oxidation, right? So that's generating these ketone bodies that occur here. So for instance, see like, let's see where. So acetoacetic acid is going to be one of those molecules that gets broken off from the triglycerides, right? That's going to end up being able to be converted over into things like beta hydroxy butyric acid, and then also acetone. So we mentioned like when someone goes into ketosis, like what does their breath potentially smell like? It's kind of like fruity, right? Yeah, so that's the acetone that's coming off of that. That's what it's able to give this that kind of fruity smell. And again, we're producing. Yes, ma'am.
So these processes, do these work similar off of like a negative feedback loop as well in the body or not?
Eventually, yeah, because the process would be that if you were able to get enough energy production, right? So say, for instance, you're kind of like in a starvation state, right? You're moving more towards lipolysis and trying to produce energy from that. Eventually, once your kind of blood sugar kind of stabilizes out, once you get to a state where you're producing enough energy, that should feed back and then inhibit further lipolysis, right? And so lots of different things can be affecting this. So for instance, if you were to have like too much cortisol in your system, right? Cortisol is the main stress hormone that we'll talk about. Like that is one thing that can generate this, right? Now you can certainly go overboard and exhaust that feedback loop and eventually have too much, kind of positive effectors causing lipolysis to occur, and this is where you can get in these states, right? So you're able to overwhelm the system, but ideally you would have a negative feedback loop that would prevent that from occurring, right? So either cortisol levels drop down, you have less epinephrine, or whatever happens to be that was originally triggering that. Yeah, good question. But again, once you have buildup of these acetoacetic acid, acetone, different things like that, this is where you end up developing that ketose, right? These are all considered these ketone bodies based off their chemical structure there, right? We can also utilize proteins for energy as well. Amino acids definitely provide a good nitrogen source as we utilize for lots of protein production and whatnot. We'll see that there are essential and non-essential amino acids. There are 8 of them that we consider to be essential. And what do you think that means?
The body doesn't
Yeah, so our body cannot produce the essential amino acids. We have to have them taken in from our diet, right? So this is like a really important thing we worry about with for the patients I mentioned who get total parenteral nutrition or IV nutrition. We have to make sure that we use protein-based products that actually contain these essential amino acids. Otherwise, if they run out of those, then they are not able to produce, like the non-essential amino acids. So they have to have it coming in from the diet there. But again, usually that nitrogen pool from the amino acids is able to be utilized for like tissue proteins, so is where you can see new growth of muscle and things like that. But we're able also to utilize some of this nitrogen here. It'll end up going into the urea cycle, as we'll see in just a minute. So I'm not going to have you memorize the list of essential versus non-essential amino acids, but just know that the differences between these two. Clinically, you're never going to have to recognize like which ones are considered essential. One thing I would note here is that histidine. Sometimes you can have ones that the body can sometimes produce, but in some cases, like children, they're not able to produce histidine on their own. Adults, they certainly could, so that would be considered non-essential. But for children, it would be an essential amino acid. And this is one of those things you kind of have to take into account when looking at differences in nutrition between, let's say, a child to an adult patient, right? They probably don't have, you'll find that especially, okay, so I don't know the exact answer to that, I'll say that, but it probably has to do with an immature metabolic pathway, right? You'll find, especially when you're looking at things like drug metabolism, that children, when they're born, a lot of things are still immature and they still take time for them to kind of mature up and to develop the enzymes they need to do certain processes, and so that's probably one of them where it takes time for them to eventually kind of be able to produce it on their own. So that happens sometimes. Yeah, we have to have it coming in from our diet.
Was there ever, because our body relies on this output that was taken, or we've always had?
Can you say that again?
I'm just like wondering, like evolutionarily, sort of like, was it losing like pathways?
Evolution isn't real. I'm just kidding. No, I have no idea. There could have been a point in our what? Yeah. Just kidding. I also don't believe in birth control. No. For the recording, that was sarcasm. No. Yeah, there may have been a point in our tadpole stage or something, or where maybe we were. No. That's probably a very good question. There's probably a good Wikipedia page out there that would probably discuss that. Specifically, I have no idea, but that's a good question. Yeah, so those may not have always been essential amino acids, but now, at least for us now, they have determined to be essential. Okay, right, so how do we eventually get some energy out of these proteins? Well, we have this process called transamination. This is primarily going to be happening within the liver, right? So you're going to see that a lot of this protein metabolism happens here. Requires vitamin B6 as a coenzyme for this to occur. And so the main ones you're going to see are going to be this alanine aminotransferase, or ALT. This is going to be utilized to convert glutamic acid or glutamate into alanine. And then you also have a liver enzyme AST, which is aspartate aminotransferase, that will convert glutamic acid into aspartic acid. So this is important to note, at least for when you do liver enzyme tests. These are some of the main ones you're going to be looking for. Right? There's some other ones that show up on an LFT or liver function test, but ALT and AST are one of the main ones you're going to look at. These help to determine, is there some damage happening to the liver to where you have these enzymes that are leaking out. Now, for instance, you typically want these values to be low. If you were to look at like a set of LFTs, if you see that they're very high, that usually means there's liver damage occurring. So even though we call them liver function tests, oftentimes they don't actually tell you the function of the liver, just tell you if there's damage happening. So for instance, when I have like a Tylenol overdose, we mentioned those hit the liver particularly hard. When they start to see that damage happening and those cells start to lyse open, they release all these enzymes, and those are the things we measure. So for instance, like your ALT or AST is probably below 30 right now. These patients can have ALTs and ASTs in the hundreds, thousands. They get very, very sick when they have a lot of that damage occurring. So those tests will come up again and again when you look at someone's liver function. But you can see this process here, these amino acids, they can be converted. Eventually they get converted over into this acetyl-CoA, and then again, when they're in that step, then they can undergo the Krebs cycle, and you can eventually get ATP being produced from there. And you can see this is a pretty complex interchange here, where you can see triglycerides, how they get involved in this process, the free fatty acids, and when they get oxidized over into acetyl-CoA, again, they can be utilized for ATP. This is also where we get, if you overload the system, that's where you get those ketone bodies, and that ketosis actually occurs there when you have too much of those free fatty acids being oxidized. And then again, glycogen and the interplay here with glucose. Again, you can see this nice cycle where if you have a drop off of one product, namely carbohydrates, you're able to kind of step in and the body can kind of shore up that energy production with using proteins and lipids. And again, this is not something we want to do all the time, but for short courses, this is probably fine for the body. Okay, so again, we have this oxidative deamination that occurs with these amino acids. This is where we can utilize some of these nitrogen sources for energy. You can also, if you have too much protein floating around the bloodstream, you can have it being stored as fat. So again, not only carbohydrates get stored as fat, but also proteins can do this as well. Some small amount of it can be stored as glycogen, or it can be degraded down to this urea cycle, where it eventually can be eliminated through the urine. Right, so urea is one of those kind of main metabolic byproducts, and the urine is going to be used for getting rid of it. Okay, so again, glucose ketone bodies mainly going to be coming out of the liver. That's where a lot of that metabolic processes occur there. So again, if you're having lipolysis and you're having fatty acids being utilized for energy, the liver is the primary thing where you're seeing a lot of that oxidation occur there. Fatty acids specifically are just going to come out of the adipose tissue, and then usually your lactic acid and amino acids are going to be utilized from the muscle tissue itself, right? Okay. And again, just another picture kind of showing the same thing, how this interplay can occur. Again, if one source is depleted, you can always utilize another one for a short period of time before you kind of overwhelm the system. Again, but the end byproduct is always getting back to acetyl-CoA, so they can go into the TCA cycle and then eventually oxidative phosphorylation. That's always the end step we get there, is we want acetyl-CoA to be able to utilize for that process. Okay. And again, looking at different sources here, so looking at the organ and how they utilize different energy sources, it's very tissue dependent. So for instance, the brain is really not able to utilize any other energy sources than glucose, right? And so someone had that question. They're like, well, what happens if you're say you go overboard on that paleo diet or something, you really have no carbohydrates being taken in? What are some of the things that can happen? And I mentioned that you can kind of look a little bit like those DKA patients because they're not able to utilize glucose very appropriately. But one of the things that takes a hit is your mental status, right? Because the brain needs glucose. If you run out of glucose, you don't have enough energy, and then you start to act a little loopy, or you get agitated, or you get very sleepy, or all kinds of things can happen to the CNS when you have no energy coming in the source of glucose, right? Again, sometimes you utilize a little bit of ketone bodies, but this is not preferred, right? Glucose is the main energy source for the brain. On the other hand, skeletal muscles, especially when they're resting, utilize a little bit of glucose. Sometimes they can utilize ketone bodies, but the fatty acids are going to be a really good energy source for kind of long-term sustained use, right? Not for kind of short burst. And then the liver is primarily going to be oxidizing these fatty acids to produce energy. But it basically can use all sources, right? It doesn't like to use lactic acid, but it can utilize that, especially that Cori cycle, to produce glucose, eventually be utilized for energy production. And then the heart, again, because we always want the heart to beat, right? If not, then you're not doing too good. But that can also, in short courses, utilize some small amount of lactic acid as well. So it depends on the tissue, where they get their energy from, and what they actually prefer, what their preferred substrates are. Obviously, with fatty acids, we get the most bang for our buck because they are able to, we mentioned, 9 kcal per gram of fat versus, carbohydrates and protein, which is only 4. So you get a lot more energy yield out of that. But again, it's not always going to be preferred, especially in short courses where you need, glucose quick, right? For whatever reason, it just does not have the mechanisms to do so, right? So you can provide fatty acids to it, but it's not going to be able to really do much with it. You're going to find that the blood-brain barrier is very selective for what it allows into the CNS, and so it's either an issue where it can't transport it, or the brain doesn't actually have the tools to actually utilize the fatty acids. But glucose, we definitely know, is going to be one of the big things that we need as a primary energy source. And this is actually one of the things we look at. So say, for instance, you have a patient who comes into the ER, you're working there, and they have altered mental status, right? So it could be anything between they're just excessively sleepy, they could be agitated, whatever. One of the first things you always check is going to be blood glucose, right? Because if that's low, we know that they need more glucose, right? Because the brain needs that, right? So it's kind of like the Snickers commercials, like someone's acting hangry, they probably just need some sugar, right? Because that's affecting the brain first and foremost. Okay. All right. So any questions on energy production, utilization? Okay, so again, no, not necessarily every step of the TCA cycle, but know kind of what we're putting into the system, what we should be getting out of it, what kind of energy yields we're getting from those, right? So kind of look at those summary slides, you can kind of see where we're going there. Realize how, you know, proteins and fats and carbohydrates can be used differently to produce energy, but again, eventually they're all going to get down to acetyl-CoA, go to that TCA cycle, right? And if we don't have oxygen, what do we do instead, right? Those are kind of the takeaway points to get from that. Okay. Okay, next we'll talk about our extracellular environment. Plays with the intracellular environment. Obviously, extracellular is everything outside of the cell. Intracellular is everything within the cell. But the question is, how do we get stuff in and out of the cell, right? So there's lots of different processes that are going to be used here. Some of these are going to be passive processes, which how much ATP do they use? 0, right? And then we're going to have active processes, which will use some ATP in order to either bring things in or put things out. Usually, you'll find that if things are going against a concentration gradient, probably going to utilize energy, right? Because we'll find that things like to flow in the direction from high concentration to low concentration. That usually is going to be a passive process, doesn't require a ton of energy, but we'll look at some of the ways that we can help facilitate that within the lipid bilayer. Okay, so looking at actual body water, you can see that we'll consider that about 67% of our body water is all contained within the intracellular space, so a pretty decent amount of it. About 33% is going to be considered the extracellular space, or extracellular fluids there, right? So it could be either in the interstitium, could be in the circulatory system, could be anywhere, but it's not within the cells directly. And find that primarily most of that's going to be interstitial fluid. So again, the space between cells is a lot of that water is going to go to, and then some of it's going to be contained within the plasma or within the central circulation. Okay, this will come up again when we talk about drugs and things like that, and how drugs like to partition out and where they like to go based on both hydrophilicity and lipophilicity. This will come up again and again in multiple courses. So again, how do we get fluids into the system? What are the primary ways we can do that? Right, so you can drink water, drink whatever is a good way to do that. Some of you guys are doing it right now. I like how I like subconsciously got you guys to all take a drink. Interesting. What are some other ways you could do it though? Say you're in a hospital setting.
Fluids.
Yeah, IVs we can do it. So certainly you give it into the veins. Is there any other ways you can give fluids? It is a process of diffusion. You can actually give subcutaneous fluids. You guys ever been to a vet and ever give fluids to your dog or cat? How do they give it? Subcutaneous, right under the skin, right? So you can actually do the same thing with people. It's a process called hypodermoclysis. So it's a fancy word you can use to impress your friends. But essentially, you can put a catheter subcutaneously. Usually we do it between the shoulder blades, and you can infuse full fluid boluses into the patient. It's good if you can't get IV access on them. It allows for kind of passive diffusion of fluids over a short period of time. But they get like a big camel back, so they kind of don't like that. But so it's a good way to get fluids in. So, and then how do we get rid of fluids? Urination is a good one. Evaporation, yes, you can have sweating is a good one. What else? Breathing, yep. So respiration, that's what do you call those losses that don't strictly occur from things like urine? It's not very sensible. They're insensible losses, right? So one of the things that you look at, especially in hospitalized patients, is you look at their I's and O's, right? So you look at their inputs and their outputs to track all sorts of things. Like, if they have edema, they probably don't have enough output and too much input, right? If they're dehydrated, they probably have too much output, not enough input. And so some of those things you can measure. You can measure urine, you can look at feces output, you can look at things like that, but you can't quantify things like how much fluid they lost through respiration or how much fluid they lost through sweating. And so we call those insensible losses, those kind of things you can't really take into account. You can't really measure very well, right? So lots of ways we can get rid of fluids, feces being another common one. I'm sure you guys run into lots of patients with things like diarrhea, with vomiting. It's another way they can expel fluids.
So you said urination is insensible?
That would be a sensible loss, because you can measure urine output, right? Either if you did with a catheter, or with children, we do it sometimes with their diapers. You can actually weigh it and get a sense for how much fluid they've lost. So those are main ways we can do that, right? So yeah, eyes and nose will be super important when you're kind of tracking fluid intake and output in your patients. Anywho, so again, once you get that fluid coming in, it's going to go into the plasma. It does not stay there. It's going to want to get into the different tissues, and so it'll be able to diffuse in a lot of cases through the capillary membranes, and then it's going to go to the interstitial fluid. We said a lot of that extracellular fluid likes to hang out in the interstitium, and then it can go bypass the cell membrane and go into the intracellular fluid, right? So this would be just strictly water getting in, and we'll look at different different types of fluids and how those can be affected based on their composition, whether it be salt, whether it be protein-based, whether it be sugar-based, all kinds of different things. But essentially, this is the process for getting fluids from the outside of a person into their cells, right? So this permeability, we know that the lipid bilayer is meant to be semi-permeable. Some things can cross very easily, some things cannot. So you think water can cross very easily? Water is actually able to freely diffuse across the cell membranes. That's actually one of those things because even though it's able to, it's partially polar, it's still able to cross that lipid bilayer. And so that's really important when we look at things like the tonicity of the fluid that we're giving, whether something is very isotonic or something is hypertonic or hypotonic. That'll be really important to see where fluid is actually going to be going through these semi-permeable membranes. But again, being semi-permeable, some things can cross and some things cannot. It depends on a lot of factors. Things like proteins, things like nucleic acids, and other large molecules. Those are things you want to keep within the cell. You don't want things like your DNA just kind of freely diffusing across the membrane. Then you can't have cell division, right? So we want those things to stay inside, and we want certain things to stay on the outside. But generally, things like ions can be somewhat permeable. Nutrients, waste products can generally be expelled with some free ability. You're going to find that it can be dependent on the cell type. Obviously, kidney cells are going to act very different than like your GI epithelia versus like your neuron. So we're going to find there's going to be some differentiation there based on the type of tissue we're dealing with. So, looking at membrane transport, we'll get into this when we talk about, get into this section in pharmacodynamics as well, so this will be a little bit of a repeat when we get to that, but we mentioned that there's passive and active transport. So passive, again, requiring how much energy? Zero energy, absolutely. So there's a few ways this can happen. It can either happen through simple diffusion, which you can see here. Right, this is just a molecule that happens to be able to bypass this lipid bilayer. It's usually pretty small molecules. They can be somewhat lipophilic and are able to cross pretty easy, no problem, right? You can also have what we call facilitated diffusion, in which you can actually have certain membrane transporters, which are either coded for a particular molecule to kind of come through. So say, for instance, you have something like, a calcium channel, or you have a channel specifically for an amino acid or something. That would be facilitated diffusion because it's having to go through some sort of intermediary there. And then some other ones will be specifically triggered by certain events. So you can have things like, voltage-gated channels, which only open when you have an active potential stimulating it, and things like that. On the other hand, you have your active transport, and again, this requires ATP to actually occur, right? So some degree of energy has to be utilized for this process to happen. And again, usually these passive transports generally happen from areas of high concentration to low concentration, right? So for instance, if I put a whole lot of sodium on the outside of a cell, sodium is going to generally kind of flow into the cell, right? Versus if I have something which is needing active transport, usually it's going to be the flip of that, where you're going to be trying to shuttle things from areas of low concentration to areas of high concentration. And because you're going against passive diffusion, that does require energy, and that's why we end up seeing ATP being used for those processes. Okay, so again, we mentioned the categories here of membrane transport. It's just passive, simple diffusion. Again, pretty straightforward there. You have the simple diffusion of lipid-soluble molecules, right? So this can be things that are able to bycross that lipid bilayer. Very, very easy for the most part. So these things should be kind of indented under the passive transport. But again, passive, simple diffusion of ions through non-specific channels, so we mentioned like calcium channels and things like that can be utilized in this process, where calcium on the outside of the cells in higher concentration than inside. You can release, once that channel opens up, calcium can flow through. Simple diffusion of water, so we just have osmosis, which is very important, especially when we look at the tonicity of different fluids and where water is going to want to flow to. And then also facilitated diffusion. So this would be something where like glucose cannot get into the cell. It's not able to cross that lipid bilayer unless it had some sort of trigger. So in a lot of cases, insulin, right? So you have glucose transporters that require insulin in order to trigger it to transport glucose, which is still in a higher concentration on the outside of the cell to go into the cell. Okay. And then active transport, we mentioned, requires that energy, the utilization of ATP. So just another picture kind of showing the process here. Again, very, very similar to what we already mentioned. This will be really, really important when we talk about different drug molecules. You'll see how they can interact with all sorts of different processes here. Usually drugs don't really interact with the simple diffusion. It may be a way that a drug can get into the cell, but you're going to find that all these are going to come up into play very, very frequently, especially when you get to pharmacology. Okay, so looking at tonicity, looking at osmosis, right? So water likes to maintain homeostasis just like our bodies do, right? So it likes to make things the same concentration kind of everywhere. Okay, I'm sure you guys are very familiar with this, right? Most, yeah, we'll go over it a little bit. We'll talk about the clinical implications of this as well, and how this is really, really important to understand for your patients. Because if you don't understand this, you can kill somebody pretty easily. So don't do that. That should always be your first rule. Don't kill anyone, right? Like, don't kill anyone and then don't get sued. Once you cover those, then you're good. I'm kidding. So anyway, so again, your molecules in a solution, they're always going to be constantly in motion. Again, they want to go from areas of high concentration to low concentration. Try to basically even everything out to make sure you have a nice homogeneous solution, right? It's going to be the same no matter which section of the solution you measure it in. Everything should be the same concentrations. So, and again, looking at, say for instance, you had had a bottle here, you have a semi-permeable membrane here. Over time, you can notice that if you have a molecule that is able to diffuse across this membrane, eventually over time it's going to diffuse, and that way you'll have equal concentrations across here, right? But for instance, if this was not permeable to this little tiny red molecule, what would happen instead, do you think? Say it was only permeable to water. Yeah, water is going to go from an area where it is less concentrated to try to dilute out the higher concentration substance, right? So you're going to see that flow of water is going to be really, really important, because sometimes we want to get water into our patient's cells. Maybe they're dehydrated, maybe they've been vomiting or having a lot of diarrhea, and they're cellularly, they're fluid depleted. We want to get fluids into those cells. Sometimes they have too much water. We want to get rid of that, right? And so we can do that by changing the type of fluids that we're giving based on the concentrations of things like salts, or changing glucose and things like that, which we'll see examples of in just a second. But again, concentration is just going to be equaling out your solute divided by the amount of solvent you have. This will be important when we talk about things like drug concentrations. So this will come up for sure when you look to see, like, how much space can a drug distribute into. We talk about volume of distribution. This will come up again. So just a little preview of that. So again, net diffusion happening here. So again, if you have something that was permeable, it goes from higher concentration to lower concentration. Again, no energy is utilized in this process. It's just natural diffusion happening over time. Pretty straightforward. Okay. Again, sometimes things can take different periods of time. Oh, yes, ma'am.
Maybe it's off topic, but like, what's the difference between, like, for fluids using LR versus natural or normal saline?
That's a good question. We're going to get to that. Just you wait. Yeah. Sneak preview. No. Yeah, so absolutely, fluid choices is super, super important for patients. So making sure you're using the right fluids for the right patient, very, very important. So we'll talk about that at length. But anyway, so that time for diffusion can differ depending on, say, for instance, if a molecule can go just straight through the lipid bilayer, it's going to be pretty fast or pretty slow process? Probably pretty fast. If it's relying on a transporter, it can be any faster or slower? Yeah, so depending on the transporter, it may actually speed the process because it might not have ever crossed on its own. But in a lot of cases, if you overwhelm those systems, if you basically take up all the spots for the transporters, you're going to have that being the rate limiting factor there. So in some cases, diffusion time may change based on how the actual molecule is getting in. So transporters can sometimes be overwhelmed, and that can slow down the process to some degree. Okay. Okay, so we mentioned simple osmosis. A lot of times you're going to find that a lot of tissues may be impermeable to water, which is good because that can help us to regulate it more effectively. So for instance, in the kidneys, it's a very good good case here. What do you kind of call this whole this whole unit here? Yeah, basically a nephron. It's kind of the main functional unit of the kidney here. It's actually the collecting duct. Here's two different nephrons I should point out. Anywho, we can utilize, usually the cells within the renal tubule are going to be impermeable to water because we want to make sure that we can regulate how much water gets into the urine, right? Too much urination, that can fluid deplete us and be dehydrated. Too little, we can get edematous and have fluid overload. We utilize these channels called aquaporins. They can be influenced by lots of different hormones and things, basically by regulating or by sensing how much water is in the system, how much solute is there in the system. You can regulate how many of these aquaporins get put into the channel and can either allow for reabsorption of water or can allow for expulsion of it. Okay, so this can be really important for the kidneys primarily, but this can also be important for things like the eyes, where if you have too much buildup of fluid in the eye, what do you think happens? Glaucoma, all right, so you have increased intraocular pressure, so you need to have water flow occur within the eye, otherwise you can end up seeing glaucoma actually develop there. So aquaporins are useful for producing the actual aqueous humor you see there in the eye. But you can also see it in the lungs, salivary glands, the brain, all kinds of different places. The aquaporins can be utilized to allow for more free flow of water in otherwise impermeable tissue. Right. Okay, so looking at the water movement, this is kind of what we were mentioning, where if you had a selectively permeable membrane or something that was not able to be crossed freely by your solute, right, or whatever is being dissolved in the solution, you're going to see that the water is going to be transferring itself to try to even out the concentrations there, right? So for instance, in this case, if you have a lot of these red molecules at a higher concentration, water is going to travel over, and so you can see that if you imagine that this is one cell, and this is the extracellular space, now that cell has less water in it, right? So you're kind of dehydrating the cell to some degree. We'll show you some pictures of what that looks like in a second under, you look at cells and their response to different types of fluids. But now if I were to have these two solutions in this jar and they were both the same concentration, how much water flow would I have? How much net transfer water? None, right? And so how would you talk about the tonicity of that fluid? Yeah, isotonic, right? So for instance, in this case, so this side A here, would this be isotonic if you see this transfer fluid? Be hypertonic, right? So hypertonic is going to lead water to flow over to it, hypertonic solutions. On the other hand, we'll say the solution here in B is hypotonic, right? So if you see no net change of fluids, it's going to be isotonic, but if you have more solute, it's going to be hypertonic. Less solute makes it hypotonic, right? So that'll be a way we talk about a lot of these solutions that we're administering to patients and how that can affect fluid flow in their cells. Okay. So hypotonic is just going to be a, so again, you have to have a comparator solution, right, to have something be hyper or hypo, right? You have to have a comparison there. So if I have solution A, and then I add a solution B, if it has less solute in it, it's less concentrated, that would be hypotonic, right? If it has higher concentration, if it had more solute in the solution, that would be hypertonic. Okay, and I'll show you some more kind of clear examples of how that will affect patients. Yeah, in this case, A would be hypertonic as compared to solution B, and that's why water's flowing to it, right? Because it's trying to dilute out and try to get an equal concentration between those two solutions. B would be hypotonic. Mm-hmm. Yep, absolutely. I promise there's going to be some clinical corollaries here in just a minute. So if you can imagine, you know, here, if for example we have you know, a semi-permeable membrane has a high concentration of glucose within this, and you have water on the outside. If you imagine, you know, this is very highly concentrated, water is going to want to flow in which direction? Into that cell. So it's going to do what? Should swell. Absolutely. So it goes. Yeah. So basically, if you're looking at the two concentrations here, you're going to see that water is going to want to flow into that membrane in order to try to dilute out that concentration. So it's going to be swelling over time. And if we look at these two different concentrations, one has 180 grams per liter of sucrose, one has 360. The water difference is going to be a lot bigger in that 360 gram per liter solution, right? Because it's going to want to try to have draw in more water to try to dilute it out even further. Okay, so that would be which one would be more hypertonic in this situation? Yeah, the 361 is hypertonic because again we have more solute in it. It's drawing more water in. It's hypertonic as compared to the 180. Okay. Okay, so looking at plasma tonicity, this is where we get to the clinical corollaries, right? So plasma has about the same osmolality, and for any of you chemistry nerds out there, osmolarity, osmolality, I'm going to talk about them interchangeably. Depending on the day, I may know the difference between the two. Most days I don't. So we'll say osmolality for this case, though. So basically, if you were to look at you know, 0.3 molar glucose or 0.15 molar salt solution, that's basically the osmolarity of plasma normally, right? We normally are going to reference this in milliosmoles per liter. And so when you measure someone's osmolarity of their plasma, this is normally what you're going to be measuring. So in a lot of cases, you'll see that 275 to 295 is considered about normal for people, right? You'll find that normal ranges vary depending on where you're working at and what lab is doing the test. So this may shift a little bit, but right around 300-ish is a normal plasma osmolarity, okay? So if I were to give a isotonic fluid, which means I give a fluid that has roughly the same osmolarity as the plasma, I really shouldn't see any fluid shift whatsoever, right? So again, the free flow of water between the outside of a cell to the inside of the cell, there should be no net change in the amount of water within that cell, right? So isotonic fluids have no net water shifting. Okay. So isotonic fluids that we give help to expand plasma volume. So for instance, if I were to give a liter of fluids that's isotonic into your bloodstream, that is going to help to fill up the intravascular space, and you don't really see any shift of that fluid anywhere. So a lot of it stays within the intravascular space, which is, you know, if you have a patient who's hypotensive, that's where you want it, right? You want fluid in the central circulation to help kind of fill the tank up, so to speak, to have the heart have something to actually pump around. So in these cases, things like normal saline. The reason why we call it normal saline is because it actually has the same osmolarity roughly as the bloodstream. And so basically, if you ever see 0.9% sodium chloride, that was considered normal saline, right? Or if you have a patient in the ER who's like seeking opioids, you tell them, hey, I got a drug for you. It's called normazoline. It's going to make you feel great. Just tell them that, and then you're good. But normal saline is very good because it is basically, if you look at the concentration, if you took 0.9 grams of sodium chloride, or table salt basically, per every 100 mLs of water. That's what that percentage means, right? So if you ever see a solution that says like 0.9%, it means 0.9 grams per 100 mLs. If it was a 2% solution, that means 2 grams per 100 mLs. That's basically what that percentage means. But basically, if you were to break that down to how many milliequivalents of sodium and chloride you have, you'll notice that it breaks down into 154 milliequivalents of sodium and 154 milliequivalents of chloride. When you add those up, that means it has a tonicity or osmolarity of 308 milliosmoles per liter. And we said human plasma is roughly what? Right around 300, right? So that would be an isotonic fluid, which means if I give you that fluid, I'm not going to cause any shift in water anywhere, inside or outside of the cells. And wherever I put that fluid, usually in the intravascular space, it's going to stay right there. And in some cases, that's where I want it, right? Sometimes I'm going to want fluid to shift one way or the other, and we'll talk about the situation where we use that. But other things like lactated ringers, this is going to be a combination of sodium, chloride, lactate, potassium, calcium. The idea with lactated ringers is it kind of looks a little bit more like human plasma, like it tries to mimic the actual ion concentrations of normal human plasma better than just table salt and water, right? And so this has an osmolarity of roughly 273 per liter, so it's still considered an isotonic fluid. Okay. Honestly, if you had a patient who's dehydrated and I wanted to give them fluids, it doesn't matter if you give them normal saline, doesn't matter if you give them lactated ringers. It just turns out that surgery loves lactated ringers. Everywhere else uses normal saline. I cannot tell you why. That is just what the medical dogma happens to be.
Well, I know, like, when we're doing, like, we have a setup, it's because it can't, the blood, if they can give that blood, they don't have to use the ion.
Okay. Yeah, so and if you look at what a normal serum sodium is, usually like 135 to 145, right? And the normal saline, the sodium ends up being like 154, right? So it's a little bit higher concentration. So yeah, that may affect it. But again, lactated ringers just more closely approximate normal human plasma than normal saline does, right? But again, they're both isotonic. You should see no shift of fluids either inside or outside of the cell, which is good for our patients. Okay, so on the other hand, though, you can have solutions which we consider to be hypotonic. And so this is going to pull water into the cell because we're giving a less concentrated fluid we're kind of surrounding the cell with that. It's going to want to diffuse water into the cell, and then you're going to see the swelling that actually can occur here. Does that make sense? So if you have hypotonic fluid, so it's going to have a lower osmolarity than plasma. So say it only has like, say 100 milliosmoles per liter, or in some cases like sterile water for injection has no osmoles per liter because it's just water, right? All of that water is going to rush into the cell, and what can happen to the cells at that point? Yeah, they pop, right? So we don't want that to happen. We don't want to have cell lysis that can occur. And so in some cases, we've actually seen patients have accidentally been given sterile water for injection into the IV, and it has killed people before, right? Not only can it lead to cell lysis like that, but it can also dilute out sodium to a dangerously low concentration, which you have to have sodium, in a normal range, otherwise you seize and die. So this is why you always really don't want to play around with sterile water, and this is why it's normally just kept in the pharmacy. You won't find it like out on the floors or anything like that. But sometimes we can give hypotonic fluids because this is good, because sometimes your cells are depleted of fluid and we want to kind of tank them back up as well. So for instance, if I were to give, say, a 0.45% solution of sodium chloride, that would be half normal saline. I can give half normal saline, and that means if I give you a liter of half normal saline, not all of it stays in the intravascular space. Some of it is going to partition out into those cells and go into that intracellular space, right? Because it is hypotonic, it's going to shift into those. And so in a lot of cases, if you are trying to rehydrate somebody, say they have really bad gastroenteritis, have a lot of vomiting or diarrhea, we will give you something like half normal saline because I know that fluid is going to shift into those cells and kind of rehydrate them to some degree. Okay. You can also have quarter normal saline, which is used occasionally, so that's at 0.225%. Less commonly, but usually normal saline, 0.9%, or half normal saline is probably the most common ones we see being used.
What would you use sterile water for?
Sterile water we sometimes use for if we're reconstituting drugs. We can use it for that. If it comes as like a dry free-dried powder, we can reconstitute it. Sometimes we will use it for just different dilutions. I'm trying to think of anything else you normally use it for. If we have to make up like our own solutions of things, so for instance, if we were making, so going back to our TPN example, that total parenteral nutrition, say we put in our amino acids, we put in our carbohydrates, we have to make up the volume difference because everyone requires a certain volume of fluid per day. We'll make up the difference with sterile water. Yep. Now, it's not the same as sterile water for irrigation. Irrigation would be on the outside of the body, and that doesn't have to be quite as regulated, right? So we use that a lot in surgery for washouts and different things like that. But this is just specifically sterile water for injections. This is a huge safety issue, which is why we just keep it in the pharmacy. Okay. Then on the other hand, we can actually have hypertonic solutions, which will draw water out of the cells, right? Because we're putting a more concentrated solution on the outside of the cells. So fluid's going to try to partition out of the cells and try to dilute out the really concentrated solution that we gave them. So basically, if you did too much, the cells would shrivel up and crenate, the term is called. We do this occasionally, and this is another very dangerous drug. It's called hypertonic saline. Usually it comes with a 3% solution, which means, we said normal saline has roughly 154 milliequivalents per liter of sodium. This has 513 milliequivalents of sodium, which means very, very highly concentrated. If you're looking at the osmolarity, it's 1026 versus our like 300 for plasma. So it's going to draw a lot of water out, which can be a good thing in some cases. Can you guys think of any examples we want to draw fluid out? Yeah, so edema might be one. So you probably wouldn't use hypertonic saline for that purpose. But where could you get too much fluid at that would be really, really dangerous? In the brain, yeah. So sometimes for cerebral edema. So if you had like a trauma patient who's having like a lot of fluid up in their brain, sometimes we'll see this with certain like DKA patients that are really, really bad. So anytime you have like cerebral edema, we can give hypertonic saline because that draws water out of the CNS and relieves that pressure, right? Because we know that can be fatal as well as having too much pressure on the brain, have a herniation of the brain stem. We don't want that. So that's the most often times where I would use it would be in the surgical ICU setting after traumas. We had a level 1 trauma center where I did my training at, and so we would use this very frequently to reduce that intracranial pressure, right? So that can be a good thing. We can do it occasionally. But normally this is another very dangerous drug where if you get their sodium levels too high, it can also end in a problem. So this is one we like to keep mostly under lock and key for the most part, unless you actually know what you're doing with it. So again, just looking at oh yes, ma'am.
Sorry, like because you said that this was for the cerebral edema. What was the other solution for
The hypotonic?
Yeah.
That would be good for like rehydrating the intracellular space. So for instance, like if I had someone who, so what typically would occur, so say for instance, I'm working at the ER at Nemours, right? And so I have a kid who's coming in, has really bad gastroenteritis, they've been throwing up for like 3 days, having a lot of diarrhea, they just look dry as all get out, right? And you guys will learn what a dry patient looks like, but they look dry. Basically what I can do is give them a liter, or well, probably not a liter for a kid, but depending on their size, give them normal saline, because that will help to fill up the intravascular space, right? So that helps to make sure their blood pressure looks good. Helps to deal with like tachycardia as well. When we get to the cardiovascular system, we'll see what that is. But then I may put them on maintenance fluids of say like D5, which we'll talk about as being dextrose 5%. So that helps give them some carbohydrates because they probably haven't been eating well. And we'd use half normal saline. And so by doing that, we're going to give a hypotonic solution that has some water flowing into the cells, and that way they can kind of rehydrate them a little bit. So that would be probably the most common cause we would do that. Other things we would do if they already had like too high a sodium concentration, we can use more hypotonic solutions to help kind of dilute that out. Okay. So again, just looking at the flow of water here between say like just a normal cell. If I were to give it a hypotonic solution, the cell is going to swell and potentially lyse. don't want that. On the other hand, if you do a hypertonic solution, it's going to cause water to leave the cell and shrink up, and then isotonic fluid should have no net change here. This picture just kind of shows the same. Basically, if you're looking at these graphs, you have osmolarity on the y-axis here, and then you have volume in liters of just fluid. So the pink area is going to be the intracellular fluid, which you mentioned is about 66% of the fluid in the body, and then about 33% of the extracellular fluid. So for instance, if I were to add isotonic sodium chloride into the intravascular space, which is still considered extracellular, notice here I only have some volume expansion here because I see no net shift of fluids. The intracellular space stays pretty much the same, right? So all that fluid I put into the veins is staying there in the veins where I may want it. On the other hand, if I were to give hypotonic solution, notice here that the fluid volume is going to be, as my osmolarity decreases, because I'm giving a less concentrated solution, you can end up seeing some water flow basically into the cells. And so you're going to see some expansion here, mostly of the intracellular space, a little bit of the extracellular space as well, right? So you're just seeing that space is just expanding out. And then with hypertonic, notice you're seeing a shrinking here. I'm increasing my osmolarity because I'm giving a more concentrated solution, and that's drawing water outside of those cells. Okay, everyone with me so far? So if I were to say, for instance, you have a patient who has too much fluid developing in the CNS, and you want to pull that fluid out, what type of solution would you give? I said isotonic, hypertonic, or hypotonic. Hypertonic, right? If I wanted to put water into a cell, I would use what type of fluid? Hypotonic. If I want to keep water right where it's at? Isotonic. Perfect. All right. So here is a comparison of some different common fluids that you would give to a patient. So you don't have to memorize this list, but these are things that will come up kind of again and again. It's probably good to know the concentration of sodium in normal saline, just because it's a good number to have on the top of your head. Especially if you have a patient who has hyper or hyponatremia, you can be affecting that pretty significantly by giving by changing the concentration of sodium that you're giving. So this is a good number to know. But yeah, sodium chloride or normal saline, this is usually the most common one you're going to see being used, and then you can see some other comparisons. So one other delineation that I'll note here is that there are two different main types of fluids. There's crystalloids and there's colloids. Crystalloids are mainly going to be made up of salts and potentially dextrose, right? So these fluids here are all going to be considered crystalloids. If you have any kind of protein that's being included in the solution, that would be considered a colloid. So you guys are familiar with the protein albumin? It's one of our main plasma proteins that helps to maintain kind of osmotic pressure within the intravascular system. But that would be a common one, a common colloid that we would give patients, being a protein-based product. You also have other products here like Plasmalite. These contain also some proteins as well. So just know the distinction between a colloid and a crystalloid, just being primarily what the solution is made up of. Oh, it's C-O-L-L-O-I-D. And crystalloid is just like it sounds, so C-R-Y-S-T-A-L-O-I-D. And again, notice the osmolarities from some of these different solutions, how these can range pretty widely depending on the solution you're dealing with. So we mentioned like normal saline is pretty isotonic. Lactated ringers, again, pretty close to that. But something like half normal saline, only 154. So again, that's going to be a hypotonic solution.
Sorry, could you repeat the difference between a colloid and a crystalloid?
So a crystalloid is made up of salts, right? Potentially dextrose as well, but mainly it's like normal saline made up of sodium chloride. That's a crystalloid, right? Your colloids are going to be anything that's made up of like proteins, right? So you think about things like albumin-containing solutions or like this Plasmalite, they contain proteins as well because they want to try to mimic some of the. So like for instance, if you have like a malnourished patient, someone who's not been eating very well for a long period of time, they usually don't have enough protein stores, right? So usually you'll see like their albumin levels drop if you were to measure it. You can use products like this to help try to replace those. Any other questions? Okay. So I think I'm going to go ahead and cut it there. We'll continue this discussion on Thursday. Any questions before I let you guys go?
Just clarify one more time.
Yep.
The last graph, table.
I can't go back because it'll screw up the recording, but you can still ask it.
Like dextrose to sodium chloride was crystalloids, and the plasmalites were colloids.
Yeah, those are colloids. Yep. Any other questions I can answer for you? Okay, it's going to be a long day on Thursday. Get 3 blocks of me. Holy cow. It's going to be fun. All right. Okay, well, I guess I will see you guys on Thursday then.